phospholipids. That is a glycerol, a 3-carbon glycerol, so a C, a C, a C, a carbon, a carbon, a carbon, and to 2 of these carbons there's a fatty acid tail. So a fatty acid at 1 carbon and another one, but then at the 3rd one there is then a little phosphate group, PO4-3 is written in the book, and at that then there is then a little other positive group containing oftentimes nitrogen. And that is now the positive part, and that can make hydrogen bonds with water. By a hydrogen bond, for example, by a hydrogen bond with water between the oxygen and the 2 hydrogens, where the oxygen is a heavier atom and it pulls stronger at the shared, in a covalent bond, shared hydrogen as the little hydrogen is just pulling at it. And so the electron is a bit pulled away from the hydrogen, and the hydrogen has lost a bit its negative electron, and the hydrogen is a bit more positive. The book leaves an open why now that positive hydrogen can team up with a positive part of phospholipid head, whatever. Now, so a phospholipid, 3-carbon glycerol, carbon, 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 and 2 of them fatty acid tails, and at the other one there is a little phosphate group, and then something with polar positive head containing oftentimes nitrogen. And they are at the cell membrane of cells, and the positive polar head turns to the water. It is hydrophilic, it allows the water to the extracellular fluid at the outside, and another row of them turns to the other side, to the cytosol, the fluid part of the cell on the inside. So they're standing then like that, and on the inside are the fatty acid tails, and then there's a polar head going to the cytosol and to the extracellular fluid. And these fatty acid tails, which can make bonds with lipids, they have oftentimes between all these carbons, then in the fatty acid tails, a double covalent bond. And then they are not together with one bond, but at that one place they are then double together, and that gives it then a little bend. And therefore they are oftentimes like that shown. And that is according to Gerard and Senegafovsky's Principles of Anatomy and Physiology book on the pages 46 and 47. But this is no medical advice and also no substitute for medical knowledge. The positions and views expressed are the ones of source and don't necessarily align with my own or with the ones of the entity spoken of. Hi, welcome. I'm trying to learn biochemistry. Today, phospholipids, and after we've done that, we're going to do Wristtraining. And if you participate in the Wristtraining, it's at your own risk. I'm just thinking, we did in cytology phospholipids, and I'm just thinking, did we do already phospholipids? But I don't think we did. I think we just did the lipid bilayer, in which we mentioned a lot of our phospholipids. These phospholipids are there with a phosphor head, which is polar, and goes to the water, and the fatty acid tails, they have 2, seems so. They are non-polar and hydrophobic, and they are then on the inside. So then all these phospholipids in the plasma membrane of cells, and the heads of them, the phosphoheads, are turning to the water inside of a cell, the cytosol, and outside the extracellular fluid. And the fatty acid tails, they don't want the water, they turn onto the inside, and that way there comes the membrane to happening. So phospholipids. Now we're having phospholipids in biochemistry. What is happening here? Like triglycerides. Okay, like triglycerides. Phospholipids have a glycerol backbone and 2 fatty acid chains, so to attached to the first 2 carbons. Very interesting. They have a glycerol backbone, like the triglycerides. The triglycerides, there was one glycerol backbone consisting of 3 carbons, and then the fatty acid tails of all the fatty acids on these 3 carbons. Now phospholipids. Also a glycerol backbone, and then 2 fatty acid chains to the first 2 carbons. In the 3rd position, however, a phosphate group, PO4-3-, links a small charged group that usually contains nitrogen to the backbone. Okay, we're going to look at the graph in a moment. So instead of having the 3 carbons with 1 fatty acid, another one, which can be different ones, we have in here also glycerol backbone, probably also at the one with triglyceride 3 carbons, 1 fatty acid, another fatty acid tail, but then the phosphate. In the 3rd position, however, a phosphate group links a small charged group that usually contains nitrogen to the backbone. So there's all of a sudden nitrogen, which is a phosphate group or something, and that is charged. So that is a polar head which is turning to the extracellular fluid. This portion of the molecule, the head, is polar and can form hydrogen bonds with water molecules. And is that why it's turning to water? It can form hydrogen bonds with water molecules. The two fatty acids, the tails, on the other hand, are non-polar and can interact only with other lipids. Molecules that have both polar and non-polar portions are said to be amphithetic. They don't say what it means. That means they say it in the other part. That means that one part is attracted to water, the other one not. As we have it now with fatty acid tails. The bilayer arrangement occurs because the lipids are amphithetic molecules, which means that they have both polar and non-polar parts. Both polar and non-polar. Phospholipids, which have this unusual property, line up tails to tails in a double row to make up much of the membrane that surrounds each cell. Okay, figure 218. C is the cell, 218C giving a 218A and C here, the cell membrane, okay? The other one here is better. And 218, okay. Phospholipids. In the synthesis of phospholipids, two fatty acids attach to the first two carbons of a glycerol backbone. Like triglycerides, phospholipids have a glycerol backbone. They never say specifically it is a 3-carbon, but they mention it like the triglycerides. That is a 3-carbon. And they explain it here. Two fatty acids, so the first two carbons to the glycerol. A phosphate group links a small charged group to the third carbon in glycerol. Is now the phosphate group linking the additional small charged group, or is the phosphor group the charged one? In B, that is here between. The circle represents the polar head region. And the two wavy lines represent the two non-polar tails. Do they now look like that, or they just represent it like that? Double bonds in the fatty acid hydrogen chain often form kinks in the tail. That is what they mean with these waves here. No, where do the kinks were previously in biochemistry? There can be something. Normally, these fatty acids, there's like that 20 carbons to the side, and each of these carbons is then saturated with a hydrogen. So we have all enough hydrogen, all these carbons, and that is then called a saturated fat. And now it can happen that such a carbon is not connected to another hydrogen, but to a carbon, and not once, as usually, but twice. It can happen that a carbon has two connections to the next carbon. And now that one carbon has not everywhere all the hydrogen, because it has instead of a hydrogen with a carbon. And now the thing is at that one place unsaturated with carbon. That is a monounsaturated fat. Monounsaturated fat. That can happen twice. That is a de-unsaturated fat. I think was that or polyunsaturated fat. I don't think. Did we ever say D? Never did it. Just monounsaturated fats and polyunsaturated fats. So more than one times there's this kink. And the kink seems to be actually there. My explanation is that the kinks comes because there is a double covalent bond. So there is the electrons of the carbons are shared, and when not just one electron is shared, and that electron is not part just of that atom, but also of that one, so we wear together, but also with the second one. And that electron is logically not coming completely over to the other one, and we're standing completely next to each other. That electron is as it is on the shell a bit further. And now we can't stay like that. They have to reach around so that they can share both. And now we're having that kink there. We're having that bend there. And they're saying that can be multiple times, so polyunsaturated, and so there are all these ways. Double bonds in the fatty acid hydrocarbon chain often form kinks in the tail. Okay, we're having the polar head, we're having a bunch of CC, and then we're having the end, that was a nitrogen. In the third position, however, a phosphate group links a small charged group that usually contains nitrogen. So it seems to be that there is a phosphate group linking the nitrogen. Polar head, we're having the phosphate group, and then we're having CC, nitrogen, CH, CH, CH3, polar head, non-polar tails, which are just the ones to the side, and then the cell membrane. Okay, phospholipids, this is a 3-carbon glycerol, are all glycerols 3 carbon, and to the first two, there are fatty acid tails, which can have at some places the carbons inside of the fatty acid tails a double bond, and when there's a double bond, there's this bend in it, and that makes the tails like that. And also the head, then is the third one, where it's not a fatty acid tail, but a phosphate group, which links it then to oftentimes nitrogen, usually contains nitrogen to the backbone, and that is now charged, and that thing in the plasma membrane are then turning with that nitrogen, which wants water and can't form, what's it on? This portion of the molecule is polar and can form hydrogen bonds with water molecules. The nitrogen part or the phosphate group part? I think because it's H3C, now it would make sense that it could make something with H2O, because it's H3. So one hydrogen goes away, it's H2, and it takes one oxygen, and now it's H2O. Oh, whatever. Okay, that is the positive part with phosphate head with the nitrogen in the goes then to the extracellular fluid on the outside and the cytosol on the inside of the cell. Now that was the biochemistry. If you prepare for biochemistry, you can go directly to the next part of biochemistry, where we will go on or back, or at one point we will read it again and make a little presentation and say what we read there about the phospholipids, or you can go also to cytology, where that is probably also important. If you stay here and participate in the wrist training, then it's at your arm rest. Go to sit stance, arms up, hands on to fists, and bring them down. There you go. That was the spotlight, now the flashlight. Fists in and out. Flat hands, the magician, to the inside and to the outside. We're having 3 rules. The first rule is that we're stopping immediately as soon as there's pain. Hands up and down, the pancake flipper. The second rule is that we're going only as far as we can go with a little bit of effort. Hands out, that T-Rex in the wall. Hands down and up. And the third rule is that we're not overdoing it. We just want to strengthen, not to exhaust. Fists and in, up, out, down. The quadrangular castle architect. And now the frog, the crab anxious to a fever. Arm up, the fist down, press on the inside. And you can take a break whenever you want. Press one up and one more down. Other hand, fist down, press. And now the arms up, the fists down into flexion, down, up, turn around. Down, up, one more time. And now the cautious big bang. The fingertips of one hand on top of the fingernails of your other hand. Open and close. Other hand. And change. Open and close. And now the Pascal bridge. Flat hands. And bend the fingers at the knuckles. Open it. Bend at the knuckles. And now the field is in Synthias director. Fists open and close. Flare the fingers. And one arm out, flex the biceps. Flare the fingers and bring it into extension and press against the back of the fingers. And again, the arm out, flex the biceps, into extension, flat fingers, press against the back of the finger. And again, you can take a break. Next exercise, the frog testing the water temperature. Hands up, down with flat fingers, up with strong fists, down. One more time. And the claw. Hands make claws and flare the fingers apart. Open. Claws, flare the fingers apart. Open one more time. And now strong fists into flexion, arm out, hands into extension, flat fingers. In strong fists, flexion, out, extension, flat fingers. And that's the last time. And now the ice axe, flat hand, bend the fingers again at the knuckles and press onto all 4 fingers. And the other hand, bend it at the knuckles and press onto all 4 fingers. And the next exercise, I think it's rocket boosters, launches or something. Long arms with strong fists into flexion. Extension, flat fingers. And flexion, strong fists, and out extension. Flat. One last round. And now go with moderate pressure over the middle part of the lower arm, the claw. And on the other hand. And that was the wrist training. I thank you very much and until next time.